For this, we'll figure it out in a minute. For this seminar, and so you can find that the handout actually is going to follow along with my slides pretty closely, so you won't have to do a whole lot of writing. When I was first asked to talk about it, I mentioned that I do have investigated other compounds other than metals, and so we included other toxicities in the title, but today I really think I'm going to just stick to metals. The metals that I'm going to be discussing are selenium, arsenic, copper, cadmium, zinc, mercury, tin, and lead. I'm using fish as a real generic term here. Most of you are very familiar with the fact that there are a lot of different species of fish, and that their sensitivities to different compounds vary greatly. And it's sort of like talking about estrogen levels in mammals. I mean, you talk about different species having very different sensitivities and different responses. Okay, let's start with selenium. Main sources of selenium are coal and petroleum combustion fly ash. Some of the products are actually reused in paving surfaces, and so sometimes they're actually reused. Glass industry is another source of selenium. Metallurgic processing, mining, electronics, scrubber sludge. There's several lakes in North Carolina that were heavily contaminated with selenium due to scrubber sludge. Selenium is an essential element. It's involved in protection of lipid peroxidation and DNA protection. Intracellularly, it's an antioxidant protecting red cells and cell membranes from free radical damage. Deficiency in fish causes mortality, growth retardation, hepatic injury, muscle lesions, and decreased glutathione peroxidase levels. Excess, a little is good, a lot is not always better. It is quite toxic to fish. The nutritive to toxic ratio is 25. You can compare that to arsenic, which is about 50 to 500, or mercury, which is about 500 to 2,000. So you can see that a little goes a long way with selenium. Excess often has the same result as too little, mortality and growth retardation. It's very stable, it's soluble at high alkalinity. There are multiple valence and oxidative forms that affect the solubility and the bioavailability of selenium. It does undergo microbial methylation similar to mercury. Small particles are often present in atmospheric pollution. Some studies in Boston have shown that these small particles can lodge in alveoli. These small atmospheric particles then can contaminate surface water and eventually get into the sediments. Selenium is toxic to fish in both the dietary and aqueous routes. It's very toxic to fish. This is the LC50 at 96 hours for golden shiners, and a lot of the LC50s that I'll be telling you are derived in very specific conditions, certain hardness, certain pHs in a certain species. And I'm sort of using fish generically, but there are a lot of differences. But you can see that the golden shiner has an LC50 of 11 parts per million. Toxicity varies with the chemical form of the compound, the species, temperature, parasite load of the animals being exposed, their age, the source of the selenium, its concentration, and the exposure length. Selenium can cause decreased temperature tolerance. For example, a species that can tolerate elevations in temperature up to, say, 35 degrees C, when exposed to certain levels of selenium, will only be able to tolerate a temperature of about 29 degrees C, a raise up to 29 degrees C. Decreased skeletal calcium levels have been noted in some fry. There have been variations in one study noted by different feeding regimes. I think it was Chinook salmon were fed contaminated fish gambusia, and they were also fed, another set of fish was fed a spiked diet instead. And the LC50s of both of those types of diets were the same, but there were changes in growth rate. These animals were carried out at different concentrations up to 90 days, and both of them had about 80% mortality at 90 days. But the fish that were fed contaminated gambusia showed decreased growth by 90 days at a much lower level of selenium in their diet than the fish fed a spiked diet. Selenium is normally found in fish. Quote, normal level found in fish is 0.47 parts per million wet weight. Comparisons between studies can be really complicated by the protocols used for determination of metals, and also just even in the reporting ways. Some results are reported in wet weight while some are reported in dry weight. In contaminated areas, levels up to close to 200 parts per million dry weight have been identified, and that sort of converts to about 65 parts per million wet weight. Hepatic levels have ranged between 6 and 34 parts per million wet weight. It tends to bioaccumulate. Water levels are less than the sediment levels, and it accumulates in plants and then more in fish. Again, when you're sampling fish, and the reports in the literature vary quite a bit of how much is accumulated in fish, some protocols have removed the gut from animals before they measure the selenium content. Others will just measure the entire fish. And if the fish intestinal tract contains a fair amount of sediment or plant material, you can get elevations in the amount of selenium identified. Accumulation in the tissues, liver is the highest, then ovaries, skeletal muscle, and testes. This varies with species again, but this is sort of a general line. Exposure type varies. Dietary accumulation seems to cause more bioaccumulation than aqueous exposure. Accumulation factors as high as 2,000 have been found in fish exposed to environmental concentrations of about 3 to 8 parts per billion. Selenium has effects on reproduction. In fathead minnows, premature hatch is noted at 15 parts per million. In contrast, zebrafish and bluegills at the same concentration have no change in their hatchability, or there's no premature hatch. At 10 parts per million, fathead minnows also don't have a premature hatch, so it seems to be dose-related. Selenium can potentiate mercuric-induced decrease in hatchability. It's caused ascites and death in zebrafish larvae at 3 parts per million. Gambusia, the mosquito fish, have shown a 41% decrease in live birth of their fry. Possible mechanisms for the embryo toxicity is that selenium may bind sulfhydryl groups on the outer membrane of the embryo, and that may cause osmotic imbalances. Also, it may cause uptake and more uptake and tissue injury. Hematologic effects have been noted with selenium. An anemia, microcytic hyperchromic anemia was noted in, I think it was bluegills. Alterations in hemoglobin formation have been reported in some studies. Dr. Ellis's study in the early 1900s found that there were abnormalities in hemoglobin formation. Dr. Hinton in 1980 reported studies with selenium on trout and could not repeat these kind of abnormalities. And when you look at both the studies, eventually vitamin E levels in the trout diet may have reduced the ability to show these hemoglobin formation abnormalities. So again, comparing between different studies can be very difficult because you may even be able to maintain the same types of environmental conditions. You may even have the same species, but if your diet's different, that can affect the study as well. Biological effects. Histopath in mammalian species, cattle have had hepatic and renal necrosis. Rats, subcutaneous and peripheral edema. Cats, cardiac abnormalities have been noted. In fish, calcification in the kidneys has been identified in trout, fed selenite, and Dr. Ferguson showed some nice nephrocalcinosis in trout yesterday. Increased renal calcium levels have been identified in other species. Proximal tubule injury, glomerular nephritis, mesangial proliferation, and periglomerular fibrosis have all been identified. And I'm lumping a lot of different studies together here, so we're mixing species quite a bit in this presentation. This is just an example of a bullhead kidney. This is a normal glomerulus, and there's very little fibroblastic proliferation around a normal glomerulus. This is a proximal tubule here. Note the hemopoietic tissue spread throughout the interstitium. This just shows an area of calcification within a tubule, and here again, you can have very, very marked calcification, and you can also have just small little tubular calci. Whoops. This is another example of calcification in tubules. This isn't quite as striking in some areas, but there is calcification in many of the tubules, and this fish was much more affected. You can see these areas here. There's also an area of tubular necrosis right here. And von Koss's stain shows nice calcium deposits. Actually, it shows calcium phosphate deposits. Periglomerular fibrosis is an example in a brown bullhead showing fibrosis around the glomerulus and also dilatation of Bowman's space. This is just another example. This shows a little neck segment coming out of the glomerulus in bullheads. These often seem to turn out of the plane of section, but they can have a very large neck segment, which is then followed by the first proximal tubules. And this just reminds you of the normal glomerulus. There's very little fibrosis. Other effects, liver, fatty vacuoles, swollen nuclei, basophilic cytoplasm, central necrosis, congestion, necrosis just in general, reticular hyperplasia, and at the electron microscopy level, alterations have been noted in nuclei, mitochondria, and lysosomes. I'm going to stand next to Dr. Ferguson on the heretic box and talk about fatty livers. There have been a lot of reports of vacuolar degeneration and fat in livers of fish exposed to multiple different types of compounds and toxicants. And I have a lot of difficulty believing a lot of the reports. There is tremendous variation in lipid and glycogen content in fish livers. Normal fish, there's a lot of individual variation, there's variation between species. So you have to really be careful when you're calling fatty change in a fish liver. This is a goldfish liver. It's, oh, wrong way. It has an area of inflammation right here. This is the intestinal tract, and here's pancreas. This liver has very little glycogen or fat. Oh, let's try the next slide. This is a higher magnification, again showing the hepatocytes containing very little glycogen or fat. This is a goldfish from the same pond, same feeding, same group of animals, neither of them exposed to toxicants. You can see there's quite a difference in the glycogen and fat content of this animal. I see a lot of variation between animals in the same group.
Sex? Sorry.
Sex, yes, sex can have a major difference as well, and reproductive status is extremely important. Females, when they're producing oocytes, certainly it's another very important variable. Also, you see that with kidney too. Some male kidneys, some male fish produce a lot of fat in their renal tubules during their reproductive season. So it can happen in multiple organs. This is a brown bullhead liver, and this amount of glycogen is frequent and fat. You can't tell without special stains which it is, but this is very commonly seen. This is just the pancreas and a macrophage aggregate. And this is totally normal. This is sort of to amuse you. This is liver. It's not lung. It's a shark, a leopard shark, and this is normal for sharks. Sharks don't have a swim bladder, and they store fat in their liver to maintain their buoyancy in the water. So if I see less fat than this in sharks, then I start worrying, because that usually means that they haven't eaten, and they haven't eaten for a long time. This is a harvest fish, and this is also normal, but this fish has very prominent sinusoids. Okay, there are true hepatic lesions. I don't want to discount all of them, but I just want to warn you that if you see a fish with some fat in the liver or fat in certain parts of the liver, don't jump on that right away as a major fatty change. This is a mummy chug, and this area has a little bit more glycogen or fat than this area here, and there's actually some vacuolar degeneration in some of the hepatocytes. This area looks fairly normal to me, and you can see here this cell I would consider being a vacuolar degeneration. There are different distributions sometimes of glycogen and fat throughout liver. There are certainly clear cell foci have been identified in fish livers. This is a herring, and you could make a case for this being a clear cell focus, a little higher magnification. And this is what the rest of the hepatocytes look like in the liver of this fish. And you can see here that there is, certainly these cells are a lot clearer, but I have seen fish that have had no environmental contamination and have a patchy distribution of fat throughout the liver, so that's not always pathologic either. Necrosis certainly is a pathological finding, and there's an area of necrosis in this mummy chug liver. And this is just a higher magnification, and you can see here that there's necrosis and cell dropout. Okay, other effects. These are mostly electron microscopy studies done in sunfish. Exocrine pancreas, decreased rough endoplasmic reticulum, increased smooth endoplasmic reticulum, increased cisternal spaces, and a possible hyperplasia. Heart. This is another one of my pet peeves. Fish do not have a true pericardial sac the way you find one in mammals. There you can have an inflammation of the surface of the heart, and I tend to call that an epicarditis rather than a pericarditis. But in this particular study, it was termed a pericarditis and a myocarditis. Gills swollen, this should be vacuolated lamellae, and I'm glad I'm not standing alone when I say a lot of these lesions are artifactual, and I would be very careful in the, when you're looking at the literature in accepting swollen lamellae as a lesion. I guess I'm sort of chauvinistic. If I've cut the gills in, if I killed the fish and I know exactly how I handled those gills, I might believe it. But if my graduate student does it, I'm not so sure I believe it. And if I find it in the literature, I really look at it with a grain of salt. Ovary necrotic follicles, that's another lesion that is also something that you can see normally with involution of ovaries. So again, you have to be careful calling that. This is just a view of normal epicardium. And this is an epicarditis. This is just artifact lifting up. I'm not ashamed to show that I have artifacts in my slides too. And this is just a higher magnification showing the inflammatory infiltrate, mostly macrophagic here, and some of the epicardial blood vessels. Gills. You saw some wonderful gill anatomy from Dr. Ferguson's lecture, and scanning EM really does show the anatomy of gills wonderfully. This is an injected specimen, so these are the blood vessels in the secondary lamellae, and you can see that nice marginal blood vessel that Dr. Ferguson was referring to, and then the see if I can get it in focus a little better, then the capillary network within the lamellus. Okay, so swollen lamellae, edema. This is the classical lifting of the epithelium from the lamellus. This fish does have some fusion as well. But again, be careful calling this lesion unless you know for sure that there has been no artifact. Ovaries. Ovaries vary a lot from species to species. Let me see if I can focus it. This is a little dark. But this is a seahorse ovary. It's one of the most beautiful ovaries that I've ever seen. And it's a nice progression of immature follicles to more mature follicles. You can see that all in one section. This is from a moray eel, just to show you a couple different species of ovaries. This is an immature ovary. And I think this is an angelfish. Yeah, I think I should turn that down. Thank you. Good. Angelfish ovary, and you can see the primary follicles and the secondary follicles. As they mature, they get larger. They contain more vitellogenin. This is from a porkfish, and ovaries. The structure changes. You know, as you saw with the seahorse, there was a spiraling. Here, you get more of a development of the follicles and filling of the center of the ovary as they develop. And these are some more mature follicles. This is also an ovary. This is what an ovary that has just spawned looks like. And it took me a while to figure out that this was ovary. Off in another section, there was one follicle sitting in one of these spaces. So that's how I was able to identify that we were dealing with ovary. So again, different stages can show different anatomy. This is from a killifish. And you can see that there is some inflammatory infiltrate here, and this is involution. And here again, you can see an infiltrate. And the macrophages will come and clean up a lot of old yolk, and they begin to look very foamy and pink. And that's normal as well. Okay, selenium, you can have behavioral changes with some of the compounds that we'll be discussing. With selenium, there's no avoidance behavior. With other compounds, you can see avoidance behavior by the fish. Anorexia is something that's been observed, and respiratory distress. Okay, let's move on to arsenic. Manufacturing, glass manufacturing is another source of arsenic. Mineral strip mines, smelting operations, electric power plants. Agricultural defoliants contain arsenicals, and pesticides. For a while in the 1940s, arsenicals were used to treat, to get rid of aquatic vegetation, so they were actively put into the water. There was also agricultural defoliants in the cotton industry. There was a fin feather lake in Texas, had a large cotton area near it, and arsenicals were used to defoliate the plants before harvest, and a lot of the arsenic got into the lake. Major fish kills, and then environmental lawsuits ended up that they had to drain the lake, dig up the sediment, bury it in clay pits nearby. So major environmental problems. There are multiple forms of arsenic. Trivalent forms exist with arsenic trioxide, sodium arsenate, arsenic trichloride. Pentavalent forms that also form arsenates with lead and calcium. There are organic forms of arsenic as well, and arsenic does persist in the environment for quite a number of years. The trivalent form is about 10 times as toxic as the pentavalent form. Smaller fish tend to be more susceptible. Organic forms are least toxic, and that's different from mercury, where you find the organic mercuries being more toxic. Oops, trying to keep in focus here. Temperature, hardness, pH, time, and concentration all affect toxicity in fish. The trivalent form, as far as accumulation, the trivalent form is accumulated more by fry. The pentavalent is accumulated more by adults. Concentration factors increase with the dose up to a point, and then you start getting the effects of toxicity, and there's less concentration. And that's shown here. You have a 0.3 concentration factor at 100 parts per million, which increases when you go up to 500, and then drops again, and that's because you're beginning to get real toxicity effects. Highest levels actually in the bile. I should have bile out here, and then liver and spleen and kidney. Excretion is in the bile, and it does undergo enterohepatic circulation. Erythrolysis has been reported, and this may be one of the reasons that there are increased levels in the spleen, because it's accumulated because of the erythrophagocytosis that goes on in the spleen. Some general biological effects, mechanisms. The trivalent form binds self-hydryl protein groups and results in enzyme inactivation and structural damage in the cell. Pentavalent form tends to affect DNA more. It inhibits phosphate insertion and also decreases DNA repair. It also induces iron storage diseases with lysosomal accumulation of iron in liver, kidney, and spleen. It's just an example of a fish spleen with macrophage aggregates, and you will see iron storage in these macrophages in the spleen. And here there's a pearl stain of liver, where you can see iron in the hepatocytes and in the macrophage aggregates. Effects on the liver that have been reported, again, fatty infiltration. Take your pick on the necrosis, central, massive, or focal necrosis. Cirrhosis, cytoplasmic vacuolation. Necrotic bodies, these by electron microscopy look like fingerprints practically. Fibrous bodies as well. Inclusions, intranuclear and cytoplasmic inclusions have been demonstrated, and arsenic has also been demonstrated to be present in the hemosiderin granules. This is just an example of necrosis in the liver. Again, this amount of fat is normal in this particular liver, and there's an area of necrosis here. Effects on the kidney: tubular necrosis, casts, glomerular dilatation. This is not a histo effect, but proteinuria and hematuria have been reported. And by electron microscopy, swollen mitochondria. That's a very nonspecific response as well. And functionally decreased respiration, mitochondrial respiration has also been reported. This is just an example of tubular necrosis in the kidney. You can see pycnotic nuclei in the epithelium. Some of the epithelial cells are sloughing off of the tubules. Mining, lead. Sources of lead, again, are mining. In Wales, in the early 1900s, there was a very bad episode in Cardiganshire where the mines produced a lot of lead, and they even have these mounds that they called gray mounds that were just, chock full of lead and caused major fish mortalities. Before unleaded gas, highways were considered a real source of lead in our environment, and there were studies which looked at body burdens of fish and correlated them to traffic density. Agricultural sources, sewage effluent, smelting, lead shot. This in the literature has sort of been attributed to feces from waterfowl, but I really tend to wonder if it's more the feces from the waterfowl or it's from the people just shooting and missing and just loading up the water with a lot of lead shot. It's very severe in benthic species that are more intimately associated with the sediments. Again, there's a lot of variation between species and conditions of exposure. Reports of LC50s vary between 0.1 and 10 parts per million. The embryo is the most sensitive stage, but all stages are affected. Smaller fish have increased toxicity over the larger fish. There is decreased tolerance with exposure to lead. In some of the compounds that I'll be discussing today, you actually can have fish that build up a tolerance to the metal. With lead, that does not happen. There's decreased tolerance. Things that have been found: growth inhibition, blackened caudal peduncles. Water quality affects the toxicity. Just the addition of 50 parts per million calcium can affect the LC50 at 96 hours quite dramatically. There are a number of technical difficulties studying lead. It precipitates in hard water. In one study, they were trying to simulate the water, natural waters, and try to synthesize them in the lab. But at the same lead concentration, the natural waters were more toxic than the laboratory waters. Lead is accumulated both by the gill and the intestine. The kidney, gill, and liver are the organs where it tends to accumulate the most. Concentration factors vary again between species, but reports have ranged between about 30 to 230. Concentration factors in liver, kidney, and gill have been reported between about 3 and 21. With time, lead does accumulate in the skeleton, and there are lead-binding proteins present in both liver and kidney. And with depuration, you see loss of lead in the kidney, but elevations in the kidney and the gill. You have depuration going on after removal from lead water, but in the liver, actually for 2 weeks, it tends to accumulate. Okay, accumulation again depends on the amount of time that's the animal's been exposed, the aqueous concentration, the pH of the water. pH 6 causes about 3 times as much accumulation versus pH 7.6. Temperature, increased temperature causes increase in accumulation, and in the literature that's pretty much related to metabolic increase at higher temperatures, and you see that in many different metal toxicities. Salinity, as you decrease salinity, you increase accumulation, and dietary calcium can also decrease accumulation. Lead is sort of unique in that the accumulation is not correlated with fish size, although the toxicity is. In many other species, it is correlated with size. There are also, in the literature, there are quite a number of problems with reported values in LC50s because there are quite a number of analytical problems associated with lead concentration determination. So some of the older literature may not be as accurate as some of the newer studies. There were actually even contamination of National Bureau standards for lead. So the older studies are still useful in that they have comparison values within their study, but the actual concentration of lead may not be exactly what they're reporting. Biological effects, hematology, there's increased erythrocyte fragility has been reported in fish. Let's see if I can get this there. Whoop. Deposits of golden brown pigment have been demonstrated in liver, spleen, and kidney, and that's presumably due to erythrolysis. Basophilic stippling, similar to what you see in mammals, and alterations in heme synthesis. It's possible that it binds sulfhydryl groups on the enzyme, causing enzyme inactivation. And this just shows macrophage aggregates with a sort of a hemosiderin type material. And, oops. And this also accumulates in the hepatocytes. You can see right here. Other effects. Histology, there's not a whole lot of histology changes with lead toxicity in fish. Paroxysmal tubular necrosis has been reported. This isn't histology, but blackened peduncles and scoliosis have been reported as well. These are just some fish to show you some scoliosis. This fish has lordosis, and you can see that sort of bent appearance right here. And usually you make your diagnosis on gross, but you can see those alterations histologically as well. There are behavioral effects. Respiration increases, and there's increased mucus at low environmental calcium levels. Neuronal and muscular abnormalities have been reported with spasms, paralysis, hyperactivity, loss of equilibrium, and lordosis. And the thought is that it's these muscular contractions that actually cause the vertebral abnormalities. There is also avoidance behavior with lead, as opposed to selenium, where you saw that there was no avoidance behavior. Oops, I think we're ready for the new cassette.
So,
Ron, have you looked for the organic form of arsenic in fish flesh?
Pardon me?
Have they looked for the organic forms of arsenic in fish flesh this year?
I know that there are the organic forms can affect fish, but I'm not sure of the levels that have been detected, if they've been detected. No. Did you have a question too?
I was just going to ask if the deviation in the fish disappeared when they were anesthetized.
I'm not sure. I don't know.
Just obviously in the acute stage of differentiating.
Right. Right. I'm not sure that, I don't remember the animals being anesthetized in those studies. Okay. Okay, zinc. The main source of zinc that I came up with is the metal plating industry. It's an essential element. It's normal in vertebrates, and it complexes with sulfur, phosphate, and phosphorus, and carbon. It's a cofactor in multiple enzymes, carbonic anhydrase in the gills and kidney. Let me see if I can. Carboxypeptidase in the gut, superoxide dismutase, LDH, phosphatase, and glutamate, which are involved in carbohydrate metabolism. Zinc toxicity has been shown in fish with mortality, growth retardation, tissue alterations, respiratory and cardiac changes, and inhibition of spawning. LC50 values vary again with species, 0.8 to 41 part per million 96-hour LC50s have been reported. With temperature, you get an increase in toxicity. The LC50s at 96 hours are the same, but the animals die faster. So your LC50 at 24 hours may change, but the overall LC50 at 96 hours remains the same. Hardness, increases in hardness tend to decrease toxicity. This is a study using fathead minnows where they examined 50 parts per million calcium levels and found that the LC50 at 96 hours is 5 parts per million, and that more than doubles when you increase the hardness. There are also increases in temperature tolerance with hard water. If you look at fish maintained at 18 degrees in soft water, the LC50 was 3 parts per million, and that drops in animals with the temperature raised. Temperature has no effect in animals in the hard water, and actually the LC50 was increased. Different forms of zinc have different effects. Zinc phosphate is insoluble and causes no mortalities. Zinc sulfate is soluble and causes toxicity. pH effects can be very remarkable. At pH 6 to 7.3 at 32 parts per million, you can have 100% mortality, which drops to no mortality at pH 8. Some of this is due to the speciation of zinc. It's free ion at less than pH 5. Increasing the pH, you get more zinc hydroxides, which are less toxic. Hardness versus pH. The effect of calcium in the water is much more important than the pH, since you, even at a pH of 8, with a low calcium level, zinc is much more toxic than even at the acid pHs with hard water. Life stage is important. The hatchlings are much more sensitive than the embryos, and those are more sensitive than the older stages. Dissolved oxygen also can increase the toxicity, low levels of oxygen. Sex, my husband says sex is always important. In fish, that's not always the case. Some males are more sensitive to zinc toxicity, but fathead minnows not so. And fathead minnows are the species that's probably the most studied in all toxicity tests. Zinc forms precipitates in hard water, and there can be real problems with experimental design when you're looking at metal toxicity. Static systems can have less zinc bioavailable because it precipitates out. It can still be toxic though. In a fathead minnow study, the authors described fuzzy fish syndrome, where there were milk white precipitates in the water, and these precipitates coagulate on the mucus, and in some theories, people have said that this coagulation of metals on mucus can actually help depurate the fish. Another theory is that just the mucus itself can cause problems with respiration. If the fish can get away from the source of the irritation to the gills or the contaminant, they can sometimes get rid of the mucus. They'll cough and get rid of the mucus. But it does also allow mechanical collection of the precipitates in the mucus on the gill, and there can be pH shifts from the gill excreting carbon dioxide, so you can actually get a small shift in pH at the gill surface, and this can increase the solubility of the metal. So some theories are that you can actually increase the exposure to the animal by having these precipitates on the mucus. But looking at the different studies, whether or not they're in a flow-through situation or a static situation can be very important, because looking at the same concentration in the same species, you can get completely opposite results using a flow-through system versus a static system. Accumulation, take your pick. Again, species differences are very important with zinc. In trout, it's been reported that there is a homeostatic control of hepatic zinc after the animal accumulates zinc to a certain level, then excretion is matched by accumulation. In red deers, in another study, there was definite accumulation in these animals. So again, it depends on the animal and the study. Accumulation is dependent on the length of time exposed and the hardness of the water. Smaller fish have increased accumulation. Zinc also affects food intake, and that affects accumulation. Now, that's not just dietary zinc, that's aqueous exposure as well. And the decreased food intake also affects accumulation from the aqueous sources. Gill is a good indicator of acute exposure. It accumulates over 100 times that of bone. Body burdens in the eye, kidney, skin, gill, and liver and muscle are in this order in freshwater fish. In your island fish, the stickleback, there is a different order. There's more in the gill and then in the liver, kidney, and gonad with acute exposures. Previous exposure also changes the concentration factor. This is thought to be due to cadmium binding protein induction in the liver, and in a study in dogfish liver, the concentration factors correlated with the amount of cadmium binding protein in the liver, and there was a lot less in the kidney. It's accumulated in scales. This is thought to be both an active phenomenon as well as the fact that these animals sometimes have decreased calcium levels because of gill damage. Retention time varies, but usually exceeds accumulation. That's different in sticklebacks. Within 4 days, even if they're not taken out of the contaminated water, sticklebacks will start to lose zinc levels. Okay, general biological effects. Severe growth inhibition. This is also thought to be somewhat nutritional. Enzyme alterations. Both increases in ATPase and decreases of ATPase have been reported in activity, have been reported in the literature. Effects on spawning. A reduction in spawning has been reported. Although the animals had spawning behavior, they never really did spawn. Histopath changes that have been reported: tubular dilatation and casts, decreased renal peritubular lymphoid tissue. This is another one of those that I'd be real careful about calling because in normal kidneys, depending on the species, just where you sample the kidney can have very different amounts of hemopoietic tissue in different areas of the kidney. So unless you know that you're looking at an entire section of the entire kidney, or you've made sure that the same area of the kidney has been sampled each time, that's a difficult one to call. Reduced adipose tissue, that's not surprising if the fish are not eating well. Retarded gonadal development, hepatocyte damage, ring nuclei evacuation, again take it with a grain of salt. Muscle degeneration and gills, lifting of the epithelium and edema have also been reported. This is an example of tubular dilatation in the kidney. In a lot of cases, when you see this kind of tubular dilatation, the animals are usually in acute renal failure, and accompanying that, you will often see an interstitial edema as well. And this is an example of lifting of the epithelium off of the lamellae. Zinc, there is a very prominent avoidance behavior at very low levels, 5.6 parts per billion. Reduced feeding has been observed, different movement. There are some really neat descriptions and experimental designs where people have made grids and used little electronic beams to evaluate the movement of animals in the water. Another thing that's been looked at is the inability to compensate in different water currents as well. And there's been an increased respiratory rate and also decreased the number of text slides in this talk, but like I said, a lot of this material is what I've collected from literature, and I don't have that many original slides on some of these metals. I will be getting to some of my own work though, so you will see some flesh. Okay, sources of cadmium, electroplating, nickel-cadmium batteries. It's found in pigments, paints, and plastics. Zinc and lead mining and smelting all have cadmium as a byproduct. Cadmium is a transitional element. It cannot be methylated, but it does form halides. It has a lower melting point, and it's more electropositive than a lot of the other transitional elements. In toxicity, freshwater species are more sensitive than saltwater species. LC50 values vary again from about 2 to 55 parts per million. Salinity increases tend to decrease the toxicity. Hardness increases, decrease the toxicity. pH, as it becomes more acid, decreases toxicity. This is different from what we saw with zinc. Temperature increases, again, increase toxicity. Species variations definitely occur, and parasitism has reported to increase the toxicity. Accumulation, the main organs are gill, liver, and kidney. There are binding proteins that hold and transport cadmium binding protein and metallothionein. There are changes in growth rate especially in acid water. Gill levels are good for acute exposures. Hepatic levels are also good for acute exposures, and they tend to indicate the severity of the exposure. Muscle turnover is slower for cadmium, and it's been proposed to be an indicator of chronic exposures. Benthic species accumulate more cadmium. Diet also affects the accumulation. It's taken up by gill and gut. It's excreted in the bile, in urine, and in mucus. And some studies have really shown that mucus is a real way for the animal to get rid of cadmium. Depuration, liver values decrease first while renal values increase, and that's different from what we saw with lead. In general, cadmium disrupts enzymatic activity. It inhibits both phase 1 and phase 2 biotransformation enzymes. It can cause vertebral abnormalities. Again, it's thought that some of this is due to muscle convulsions. It causes increased mucus production, which has been reported to cause anoxia. Hypocalcemia has been reported in fish, and that's been attributed somewhat to gill damage. Low collagen and low calcium levels have been reported for some species and in the vertebrae, and have been, they think that the low calcium and low collagen are why the vertebrae themselves are weak. Again, that's species specific. Some species have no changes in calcium levels of the vertebrae. Reproduction, decreased embryo survival occurs at 80 parts per billion. Congenital defects have been reported. Hematologic abnormalities, red cell abnormalities reportedly due to alterations in RNA synthesis. This is, again, an accumulation from a lot of different papers. So some of these things have been found in some species, but not all of them in every species. Hypocalcemia, hypokalemia, hypermagnesium, hyperphosphatemia, hyperglycemia, hemodilution in freshwater fish, and hemoconcentration in saltwater fish. This is another point here that I want to talk about, and that is the effect of compounds on packed cell volume. You'll find in the literature that people will report packed cell volume drops, and that this is a definite anemia, and yet when you look at the article you never find that they did total protein evaluation of the serum or looked at the osmolality of the serum, and that's incredibly important. If you have a compound that affects renal function and you get in a freshwater fish, which is basically a little bag of electrolytes floating in a big pool of water, and water is constantly trying to enter the fish. If your kidneys are not working and you're a fish in fresh water, you're going to blow up like a balloon, and that's where you get the picture like Dr. Ferguson showed you with the raised scales and ascites and exophthalmia, because they're basically water overloaded. This is going to drop PCV, and it's going to drop it dramatically, and unless people are looking at total protein or osmolality of the serum, they're going to say, hey, these animals are anemic. Saltwater fish have a different problem. They are constantly trying to retain fluid. As a matter of fact, renal anatomy changes quite a bit in saltwater specimens. Some fish have actually eliminated glomeruli, so there is no active filtration. Anything that gets out of the kidney goes via tubular secretion. Other fish, other saltwater fish also have eliminated distal tubules. So they basically are proximal tubules with or without a glomerulus connecting to a collecting duct. So again, any compound that affects renal function can affect what you're seeing as far as PCV. Okay, histopathology. Intestinal mucus cell hyperplasia, that's one that, again, I'm not so sure about, but it has been reported in the literature. And epithelial swelling. Renal changes, proximal tubule necrosis has been well documented. Cast formation, hyaline droplets, dilated tubules, and reduced brush borders, which often is a sublethal change in proximal tubules. In the testes, reports of hemorrhage and necrosis, gills, hypertrophy, hyperplasia, mitosis, which goes along with the hyperplasia, and in the heart, pericardial edema. This is just an example of hyaline droplets within proximal tubules. This is a brown bullhead. Hypertrophy of epithelium. One thing that's been reported in literature a lot of times, this I just sort of threw in, people have reported in various toxicities an increase in mucus cells along the secondary lamellae. I don't see that very often. This is sort of an unusual finding for me to find this. But what I usually find is if the fish seem to have a lot of mucus in their gills, histologically I don't find any mucus cells, and I think it's because they tend to expel all of their mucus, and so they're smaller and you don't see them very well. This shows fusion at the tips of lamellae and hypertrophy of the epithelium, and also some ratty edges there, a little bit of necrosis. Again, hypertrophy, you can see here, the epithelial cells will become sort of a glassy appearance and pale eosinophilic. And that is how chloride cells are described in a lot of fish. And frankly, without an electron microscope, I have trouble sometimes deciding whether something is a chloride cell or a hypertrophic epithelial cell. So chloride cell hyperplasia, again, is one of those lesions that I'd be careful about calling. Okay, also with cadmium, abdominal edema in the muscles has been reported. This is just an example of normal muscle, and this is edema. Behavioral effects, increased respiration, a sneeze-like response. Sometimes the fish will sort of move forward and backward and wiggle their operculum. Narcosis has been reported. No avoidance behavior. Okay, copper. Now I have done some studies with copper, and I'll be showing you some of my experimental data here. Copper is an essential element like zinc and selenium. It's involved in enzymes and glycoproteins, hydrogen peroxide destruction, energy production, iron absorption and transport, myelin maintenance, bone, brain, and hemoglobin synthesis it's important in, and it's bound to ceruloplasmin or alpha globulin in the blood. Its toxicity is complex. Again, there are species of copper, and they have different toxicities. The copper ion is very toxic, more so than the hydroxides. pH affects what species you have in the water. There's increased ionic forms at the lower pHs, increased carbonate and hydroxide forms at higher pHs, and copper precipitates out a lot of times at higher pHs. Hardness also can precipitate out copper, and even at low pH, copper can be chelated with high levels of calcium. Organics tend to decrease the amount of free copper ion, and therefore decrease mortality. Organic material, though, in a contaminated site can be a reservoir for copper, and with decreases in pH or changes in the environment, you can then get a release of some of this ion from the organic material. Salinity increases cause a decrease in mortality. Temperature, again, you've seen this before. Higher metabolic rate causes increase in mortality. Smaller fish tend to accumulate more, and that's thought to be due to higher metabolic rates. It inhibits sodium-potassium ATPase, and reports have indicated that some fish species can lose about 50% of their sodium. Some tolerance exists with copper toxicity. It's usually about 1 week. So if a fish has been exposed to copper, it may be more tolerant if exposed again within a short period of time. There's tremendous species variation as far as copper accumulation. Some of you may be familiar with Dr. Fraser's and Bunton's work at Hopkins with the white perch from the Chesapeake Bay. They accumulate large amounts of copper in their liver. These are hepatic levels, and the striped bass is sort of the reference fish here. There appears to be no apparent problem with these fish. It's interesting, in some of the fish that have had hepatic lesions, foci of alteration, those areas don't take up the copper, but the rest of the liver does. Mucus chelates copper, although it is ineffective at low pHs. Hepatic accumulation is time and concentration dependent. Liver is the major organ for accumulation, then the kidney and gill, brain, spleen, and gonad. General effects, metallothionein is induced, and that may again be one reason why there is some tolerance. Ammonia secretion raises the pH and can be somewhat of a protective mechanism. Prolactin levels go up with copper exposure, and that can enhance mucus secretion and chelation. Again, it inhibits sodium-potassium ATPase in the gill. Reproductive effects have been noted with copper: decreased hatch, embryonic deformities, reduced yolk absorption, and decreased egg production. Hematologic effects have been noted, and in the literature you'll find reports that say there are increased packed cell volumes and other reports that claim decreased packed cell volumes. And again, a lot of these reports don't talk about total protein values or what word am I looking for? Osmolality of the blood, yeah, thank you. So there are osmoregulatory changes that have been reported as well. Histopath, lesions have been noted in the lateral line mechanoreceptors, olfactory chemoreceptors, vascular necrosis has been reported, gills, necrosis, hyperplasia, inflammation, liver necrosis, and kidney necrosis of hematopoietic tissue. This is a lateral line and showing the mechanoreceptors. And this is a lesion in the lateral line showing spongiosis and a necrotic epithelium. This is to remind you that I have done some work with copper. This is a large diluter system that we have in the lab. And take a look at the color of the tanks. These are very light polypropylene tanks. These are brown bullheads, and the control fish had the ability to light adapt to these tanks. If you keep them in a dark tank, they're dark, and if you keep them in a light tank, they get lighter. This is a fish that was exposed to copper and was maintained in one of these tanks as well, and you can see that it had lost the ability to light adapt. Okay, this is nasal epithelium, and you can see that it's ciliated. And in fish that we exposed to 100 parts per billion copper in the water, you can see that in some of the animals we found loss of the ciliated epithelium. That's a sublethal damage, and in my mind I sort of correlate it to something like the loss of the mucociliary apparatus in smokers. This is a brown bullhead kidney again. This is that neck segment that I was talking about. The glomerulus is somewhere up here, either in or out of the plane of section. The neck segment is ciliated, and you can see these long cilia running through the lumen. This is a proximal segment. We found hyaline droplet formation in proximal tubules. You can see the brush border here. And this thin line here is actually a line of desmosomes. You can sort of see that pinkish line in the front. Electron microscopy, this is a toluidine blue section. I did electron microscopy in these, and you can see these hyaline droplets really nicely. This is a protein cast in a distal tubule. By electron microscopy, they're just dark blobs within the cells. And I did elemental analysis thinking, oh, right, we're going to find copper, but we didn't. It's probably just protein that's been reabsorbed from leakage through the glomerulus. With elemental analysis, you have to be careful not to use copper grids if you're looking for copper. We used nickel. You can also use nylon grids. These are control gills in the bullhead. And this is what we found with 100 parts per billion. There was a very intense inflammatory response. You have to be careful with gills. Sometimes sectioning artifact can give you an area that looks sort of like this. But if you're seeing the individual lamellae showing up here, you know that that's not sectioning artifact. And at first I really didn't believe that this is what I saw, and I went back and I had them recut and recut and recut, and it was this way all the way through the gill. And you can see there's quite an inflammatory infiltrate. And here you can see the vascular outline of the lamellus right here. Did a second experiment where I was exposing at a lot lower concentrations, 25 parts per billion, and this is at 96 hours. A lot less inflammatory component here, but a definite hyperplastic response. These cells also look larger to me. They get a more squamous appearance. In addition to looking at the effects at 96 hours, I took the fish and put them in fresh water, and within just a few days you can see, this is one day, you can see that this epithelium looks a lot better. So they have the capability of repairing. This is at 2 days following removing from copper. And you can see there's still fusion, there's still pathology here. But a few of these lamellae look pretty normal. And certainly the fish is able to breathe a lot better with lamellae that look like that. This is 5 days following removal from copper. And you know, there's still pathology. You know that these gills have had some kind of an insult, but there is a remarkable capacity to repair. So we only had limited, this was part of a different study, and I only had a few samples of fish, so I decided to really look at this in some goldfish. This is just a goldfish gill. And within 48 hours, you can see there's a tremendous hyperplastic response in goldfish. This is at 55 parts per billion. And in addition to fusion between the lamellae, you also had fusion between the filaments. And you know, this is not uncommon in the sections. That was at just 2 days. This is a control gill showing bromodeoxyuridine incorporation, and you can see that there are scattered cells that have taken up the label. Bromodeoxyuridine is an analog of thymidine, and it's incorporated into cells during the S phase. So we're not seeing division here, we're seeing cells that are getting ready to divide. And at 48 hours, you can see that there is a definite increase in the number of labeled nuclei, as there is also an increase in the number of nuclei, period. But there is definitely a proliferative response going on here. This is just a higher magnification. You can see that a lot of these cells, this is, you can't see the cytoplasm here, but the cytoplasm is very big compared to the regular cytoplasm in a normal epithelial cell. And I think part of the reason that recovery may be so rapid is that you may actually not lose as many of these cells, as some of them may just shrink down in addition to eventually losing them.
What kind of cell?
These are epithelial cells. The flat epithelial cell that lines the lamellus.
So respiratory cell.
Respiratory epithelial cell.
Is inner or outer?
That I can't tell you because we've lost the.
Is that also part of progenitor cell?
Well, yeah, it's hard to say. I mean, these cells are mitotically active. You can see they're mitotically active out here. Now, I would assume that these would be, the first layer of cells that would be dividing would be the outer layer, but I haven't really been able to differentiate whether this is in the inner or outer layer. And at this point, you know, you can't really tell where it would have come from. Oops, did I just go backwards? Okay, this is at 96 hours. And you can see again that there is not only fusion between the lamellae, there's fusion between the filaments themselves. These fish are up at the top of the surface of the water gulping for air. And you see proliferation down at the base of the filaments, of the primary filaments, and throughout the entire lamellar structure. This is at 96 hours. So you can see that there are a lot more labeled nuclei at this point. Normally, you tend to have epithelial cells in the normal gill be proliferating here, but I have seen cells, as I showed you on the control fish, cells even up here along the lamellus will also be dividing. So they are active along the entire lamellus. And this is just another example showing you also get this sort of entrapment and some inflammatory infiltrate in here as well. This is just a higher magnification showing you the proliferation, and this is between two primary filaments, the fusion. Okay, and this is just another control, and as I said, you see cells that are proliferating out here in the lamellus as well as down here. So you do, it's sort of like the epidermis in the skin. You have proliferation at multiple levels. Okay, this is 2 days following removal of the copper, and you can see that there's a shrinkage here of this proliferative epithelium, and you're beginning to see some of the tips of the lamellus. And this is another section at 2 days, and this one you can see that there's actually even more recovery in this fish, and there is a lot less proliferative activity going on in these gills. This is at 7 days following removal from the copper. And the repair process continues. You can see, I mean, you definitely know that these gills have been hit, but they look a lot better than they did at 48 and 96 hours. I actually did some counting and morphometry on these and came up with some numbers, and the control numbers of nuclei were fairly constant, and at 2 and 4 days of copper, and even 2 days after copper removal, you still had much increased over controls number of cells. And, oops, sorry about that. And when you look at labeled nuclei, it's even more remarkable. Certainly by 2 days, you've really had a major proliferative response, which continues up to the 4th day, and then rapidly drops off as soon as you take the fish out of copper. Okay, other biological effects, there is avoidance behavior, and as I mentioned, there's increased gilling, and they're at the surface of the water. Okay, yeah?
When one treats with copper sulfate,
Yeah.
what kind of concentration?
If you're treating with copper sulfate, are you talking about a saltwater tank, or are you talking about ponds, freshwater ponds?
Well, I think it may be a aquaculture rather than freshwater.
Freshwater. Okay. A lot of times you'll have algal blooms, and people will treat, and they will treat with higher concentrations than I've used in the lab. But as I mentioned, there's a lot of organic material in these ponds. So, but I think that you can definitely get toxicity at levels that are used in the environment. That's for sure. And I think aquaculturists will sometimes overdo it. Any other questions on copper? Chemical plants, oh yeah, I never did give you sources of copper, and that is one of the major sources, is putting copper into aquaculture or into even ponds or lakes to cut down on algal blooms. Copper paints also are anti-fouling paints, are used in boats. And since tributyl tin oxide has been banned in several states, copper may be more of a factor. It's also used in wood preservatives. Okay, chemical plants, sources of mercury. Certainly the Minamata episode where they used mercury as a catalyst is one of the major environmental disasters that happened with mercury. It's used as a slimicide in pulp mills. It was used as a fungicide in agriculture, and it's also used in plastic production. Methylated forms are the most bioavailable. It is accumulated from the diet and the water, and there, again, it depends on if it's a benthic or pelagic fish where they accumulate more of their mercury from. There's a lot of individual variations. Benthic species tend to accumulate more than pelagic species. Older animals tend to accumulate more over time than younger animals, and females tend to accumulate more than males. Levels in tissues, kidney, more than liver, more than muscle, more than bone, and again, that's species specific. Ranges, very big range reported in the literature, from 11 parts per billion to 1,800 parts per billion. The chemical form is important. Methylmercury is much more toxic in fish. Specimen size is important. Smaller fish are much more sensitive. It ranges between 3 and 10 times more sensitive. Temperature, again, increased metabolic rate. In one study where they measured metabolism and said it was about twice, they also found that there was about a doubling in the toxicity. Hardness also affects mercury chloride or mercury toxicity, and the youngest animals are the most sensitive. General effects: mucus binds free mercury better than it does methylmercury, and methylmercury is the most toxic form. Gills accumulate the mercury first. It's demethylated at the gill. It's bound to metallothionein, transported to the liver, and some species remethylated in the liver. And again, methylmercury is twice as toxic to gills. Hematologic changes have been reported in the literature. Erythrocytes, vaculation, and microcytosis. Neutropenia has been reported, as has thrombocytosis. Histopath, necrosis in multiple organs. Gill, different things have been reported about gills. Necrosis, ballooning, degeneration. Again, take that with a grain of salt. Mucus, hypertrophy, hyperplasia. Pancreas, duct alterations, vaculation, necrosis. Liver, vaculation, necrosis, and regeneration. And kidney, necrosis, and calcification. I've used mercury as a toxicant in goldfish kidneys, and I have to admit I was not looking at it as an environmental contaminant. I was looking at repair responses at sublethal doses in fish, so I was using it as a model, and I injected at IP, so it's not a very environmental study here, but it does show you some of the effects that toxicants can have on fish kidneys. That's a normal kidney. This shows an area of tubular necrosis following 5 microliter per kilogram IP injection. This is a glomerulus here. Distal tubules were not affected. A few days down the road, you see casts and some regenerative attempts along the injured nephrons. Whoops. We may not be able to see the regenerative effects. Oh, it's the next carousel. We'll see it. Thank you. You can see here a flattened basophilic epithelium lining these dilated tubules and these are proximal tubules that have been injured. You can see the glomerulus again, not much injury there. This is similar to what you see in rats with mercuric chloride poisoning. Bromodeoxyuridine, you can see that these flattened basophilic cells also incorporate bromodeoxyuridine. Now, one thing that I've found in my studies of fish, and I've looked at a number of different species and a number of nephrotoxic compounds now. The goldfish is the one that I'm the most familiar with. I have seen the development of new nephrons following nephrotoxic injury, and that is a response, as far as I know, unique to fish. These developing nephrons are very similar to what you see in mammalian embryos and also in larval fish. Fish retain the ability to produce new nephrons throughout life because they grow throughout their entire life. So it's okay to see an occasional new nephron in a fish. Don't assume that when you see one, you've had a major episode. But if you see a lot of them, then you should be looking at the kidney and saying there's something probably hit this kidney if it's an older fish. You can see this is a glomerulus, this is a mature glomerulus, and right here you can see immature glomeruli forming in these structures. These show up, it depends on the toxicant and it depends on the fish species. In goldfish, following hexachlorobutadiene, I see these developing at about 3 weeks. With mercuric chloride, they came up at about 2 weeks. But it is a response that happens well after the toxic insult. And to some degree, I think that it is a possible use for the developing nephrons in fish, wild fish, as markers of nephrotoxic damage in the wild, especially if you have an intermittent exposure. You may not always have a massive die-off. This is a sublethal to the fish injury, and yet you can see major changes occur in the kidney, and you can see that over a period of about a month or more even. So these are these developing nephrons. You can see a developing glomerulus here. A lot of these also have mitotic figures. This shows an older glomerulus, and this one, I don't think that the tubule that it's attached to is probably very healthy because we're beginning to get some fibrosis, and the tubules that do not repopulate and regenerate will often eventually show scarred glomeruli or periglomerular fibrosis. So here you can see the podocytes developing, and you can see mitotic figures.
Can you see nephroblastoma?
I have not generated nephroblastomas, but that is a plan of mine. One, to produce new nephrons in a series of fish where we have a whole population of more or less synchronously developing nephrons, and then in an adult fish then expose it to compounds like ENU, which have been shown to produce nephroblastomas in rabbits. And as a matter of fact, I did a preliminary study with just a few fish. It caused a fair amount of necrosis in the kidneys with ENU, but I haven't really done a good experimental study looking at production of nephroblastomas. I have seen nephroblastomas in wild fish, not real frequently, but I have seen some. This is one of these developing nephrons, and you can see that, again, bromodeoxyuridine incorporation is pretty prominent. This is another one that's a little bit older. As these nephrons mature, the tubules get longer. These little small clusters that I showed you before get bigger. As the cells differentiate, they get more eosinophilic and eventually become normal tubules. They penetrate existing collecting ducts, and I don't have one with me, but I have some good shots of actually where you can see these things penetrating into the collecting duct, and that's not a real surprising finding because that's also what happens in the embryo, especially in mammals where you have the metanephric kidney where the ureteric bud grows up from downstream and starts inducing blastic epithelium to form tubules, and those then penetrate into the ureteric bud and the collecting duct epithelium. Okay, other effects of mercury, behavior changes, vision impairment, and swimming disorders have been noted as well. Okay, tin. This is the last one that I'll be talking about. Sources of tin: anti-fouling paints, hospital disinfectants, molluscicides, preservatives of wood, cotton, textiles, and paper. Tin plating, solder, brass, and food packaging are all sources of tin. The organic tins, forms that are found in salt water: tributyl tin chloride, tributyl tin hydroxide, hydrated tributyl tin cation. It accumulates in the water surface microlayer. As a matter of fact, in the very thin petroleum film that's on the surface is where you can find the majority of the tin. In marinas, you get a mixing of the surface layer, and there's been a range of what's been found in the marine environment. Smaller fish are more sensitive to tin. Again, there's species variation. If you look at rainbow trout versus lake trout, there's difference in the LC50s. Age is another factor. The larvae are much more sensitive than the subadults. Decreased hatchability has been noted in some species. Mechanisms, possible mechanisms: inhibition of lipooxygenase, possible inhibition of oxidative phosphorylation, and there have also been decreases in P450 activity noted. Bioconcentration, you've heard this before with all the other compounds. It's species-specific. Half-life is also species-specific. Tributyl tin is lipid-soluble. In bivalves, concentration factors, incredible concentration factors have been found, and there are apparently no enzymes to degrade the tributyl tin. In fish, tributyl tin is degraded to dibutyl tin, to monobutyl tin, and then to tin via P450. General effects: loss of equilibrium, erratic swimming, lack of weight gain, avoidance, again, concentration dependent and species dependent. Hematological parameters: decreased hemoglobin concentration and decreased hematocrit have been reported. Histopathologic effects: in the spleen, lymphocyte depletion, reticular endothelial proliferation, lymphoid karyorrhexis, and erythrophagocytosis have all been reported. Liver, biliary necrosis, peribiliary hepatocyte necrosis. This is one where I see red flags popping up when I read that, because again, fixation can really affect this. If you don't get those tissues into fix right away, it's a very easy artifact to see. And again, vacuolation has been reported. Okay, this is a spleen of a gray angel, just to give you morphology of spleen. This has a very prominent ellipsoid structure, and you can see the macrophage aggregates. Here again, the ellipsoids in this species are really prominent. And that's for people that aren't used to looking at head, kidneys, and spleen. Unfortunately, I didn't bring a head kidney, but look for ellipsoids if you're trying to figure out if you've got head kidney on a slide or if you've got spleen, because sometimes it just looks like a wash of hemopoietic tissue. So sometimes if you can find one or two of these in a species that doesn't have as many, you know that you're in the spleen. Some species also have very prominent nerves in the spleen. Okay, this shows lymphoid depletion and also shows large macrophage aggregates with hemosiderin-type pigment in them. And you can see that there's a very prominent reticular endothelial pattern here, and just not very many lymphocytes at all. Again, right here. Okay, other histopathologic effects: gill necrosis, inflammation, vacuolation, and edema. Pseudobranch, vacuolation and inflammation have been reported, and kidney necrosis, vacuolation, and hemosiderin and macrophage aggregates. Other effects: thymus-dependent, species-dependent atrophy. There you have to be careful about age as well, because thymus does atrophy with age. In larvae, skin and muscle lesions have been reported. In the eye, degeneration of lens, cornea, retina in larvae, and CNS vacuolation in larvae. And it'd be interesting to have a neuropathologist look at those slides. This is just an example of a fish eye. As you know, they have round lenses, cornea, spaces of Fontana, retina, choroid. A lot of fish have different varying amounts of choroid, and in some species there is a vascular connection between the choroid and the pseudobranch. Okay, this shows a degenerating lens, and I have a higher mag of that. This is a fairly normal lens with a normal nuclear layer. And you can see in this fish the nuclear layer is necrotic, as is most of the lens. Gill pathology has been documented. Hemorrhage has also been documented, and thrombus formation. And again, you can create hemorrhage in the gills by increasing the pressure over the head by karate chopping the fish. So that's one thing to watch out for when you're reporting hemorrhage in the gills, or telangiectasia, as this is called. But if you find things like this, which is thrombosis, which is what was in those gross samples, you know that happened, that hemorrhage happened ante-mortem, or prior to when you had your hands on the fish. And you can see typical lines of Zahn in these thrombi. So lastly, you can see gill edema in tributyl tin oxide exposures. But again, that's one of those lesions that you want to be very careful when you report. It's a higher mag. And that's it. So I've given you sort of a whirlwind tour of metal toxicity, and I'd be glad to entertain questions, but remember, I'm not an expert on metals, but I've tried to give you a good review of some of them. Yeah?
How about the zinc pennies we have now? Is that zinc very soluble, like throwing the pennies in the wishing wells?
Yeah, I think that people have voiced concern about children eating the pennies and getting zinc toxicity from that. And I'm not sure, but I think people have mentioned that it does dissolve in GI tract of mammals. Now, I'm not sure about fish, but certainly just throwing copper into the fish ponds is a real problem, and educating the public is important. And yet, I was at a zoo just the other day, and I mean, there's this big sign that says, you know, don't put coins in the water, and there's tons of coins in the water. So it is a problem, and zinc toxicity may actually be more of a factor in the future. Yeah?
Comment about mercury. Another source of mercury that we experience in the western part of the United States is natural mercury, and that comes up particularly in the area I live. It's out of the coastal range of California, where there's a lot of volcanic activity, and hot springs in which the mercury will come up, and there's a number of lakes there in which pregnant women are warned against eating any fish out of there, and I think others one fish a month or something, one meal a month out of fish in those areas. So there's mercury, because of its natural situation of being a liquid, can move about much better than the others, and can come up through the grounds, and it's not necessarily
Anthropomorphic,
associated with, activity.
Anthropogenic.
It's natural.
Yeah, yeah. And that's with selenium too. You know, there are lots of natural sources too. I think I focused on contaminant sources. But you're right, and I know I went fishing in New York a while back, and I was reading my new fishing license, and they say, you know, pregnant women shouldn't eat any fish out of New York State. And they also said there were a lot of limits on the size of fish that you could keep. And they, you know, a lot of them were 12 inches. You had to throw everything back that was under 12 inches. And then they had a health advisory, and they said, eat fish, don't eat any fish that are bigger than 12 inches. So I mean, I was saying, well, we can only keep the ones that are 12 inches or bigger, and we can only eat the ones that are 12 inches or smaller. So basically, don't eat fish, but you gotta live. You gotta eat fish every once in a while. Okay, thank you.